This video will take a look at pyruvate oxidation and the citric acid cycle. Recall that this is stage 2 of cellular respiration. We just finished glycolysis, which was stage 1. At the end of glycolysis, we have 2 pyruvate molecules. We took a single glucose, which was a 6-carbon compound. We broke it into 2 3-carbon molecules, which are the pyruvate. This all occurred back in the cytosol. So glycolysis occurs in the cytosol. Now we're going to take pyruvate and we're going to ship it into the mitochondria, where the rest of cellular respiration will occur. So stages 2 and 3 will happen inside the mitochondria. So we're going to take pyruvate into the mitochondria, and then that's where we will run stage 2. The first part of stage 2 is pyruvate oxidation, where we will take this 3-carbon compound of pyruvate and we will transform it into acetyl-CoA. Then we are going to take acetyl-CoA and run it through the citric acid cycle. During the citric acid cycle, we'll continue to break down our hydrocarbon, we'll produce ATP, and we'll also produce some electron carriers, NADH and FADH. So let's take a look at those steps in a little more detail. The first part was the pyruvate oxidation. Recall we had a 3-carbon compound at the end of glycolysis. We're going to bring it into the mitochondria here through a transport protein. Then we are going to convert that 3-carbon compound into this 2-carbon compound. We will use this enzyme, pyruvate dehydrogenase, to do this. The pyruvate dehydrogenase will remove CO2, so we're removing this carbon dioxide from this 3-carbon compound. That's why we're left with 2 carbons over here. And at the same time, we're also going to give a hydrogen to NAD to create NADH. Remember, this is an electron carrier that we can use later to generate ATP. So after we release the CO2 from our 3-carbon compound, we're left with a 2-carbon compound. Then we attach that 2-carbon compound to coenzyme A to form our acetyl-CoA. The acetyl group is this 2-carbon group, and now acetyl-CoA has it attached to coenzyme A. Now remember, there were actually 2 pyruvates over here for every molecule of glucose. Glycolysis started with a 6-carbon compound and ended with 2 3-carbon compounds. So we really do this twice if we're tracking 1 molecule of glucose. If we were tracking a molecule of glucose, we actually have 2 acetyl-CoAs over here, and then we have 2 carbon dioxides being produced here, and we have 2 NADHs being produced here for each molecule of glucose. It's easier sometimes to just keep track of a molecule of glucose. Let's take a look at this video that animates this for us.
Let's follow this pyruvic acid molecule into a mitochondrion to see where most of the energy is extracted. As the molecule enters the mitochondrion, 1 carbon is removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA.
The product of glycolysis. The pyruvate molecule is the source of carbon and electrons for aerobic respiration reactions inside your mitochondria. Pyruvate enters pores on the mitochondria membrane and is transported into the interior. The reactions linking pyruvate with aerobic respiration are performed by a huge protein complex made with multiple copies of 3 types of enzyme. The first enzyme catalyzes decarboxylation, transforming 3-carbon pyruvate into a 2-carbon acetyl group, generating carbon dioxide as waste. The acetyl group is then grabbed by the second enzyme, using flexible arms to efficiently transfer the product between reaction sites. In the active site of the second enzyme, the acetyl group is attached to coenzyme A, generating acetyl-CoA, fuel for the citric acid cycle. Two electrons from pyruvate oxidation are retained by the second enzyme before passing them to coenzyme NADH, catalyzed by the third enzyme. Coenzyme NADH travels through the matrix, delivering electrons for the electron transport chain. In a sequence of reaction steps, the pyruvate dehydrogenase enzyme complex generates fuel for aerobic respiration reactions of the citric acid cycle and the electron transport chain.
Now that we've formed acetyl-CoA, we're ready to enter the next part of stage 2, which is the citric acid cycle. So we'll take acetyl-CoA from the first part of stage 2 and send it into the citric acid cycle. Again, this is happening inside the mitochondria. Let's take a look at the overview of the citric acid cycle first. This is also sometimes called the Krebs cycle from the person who named it, and it's sometimes also called the TCA or tricarboxylic acid cycle, but more commonly now it's referred to as the citric acid cycle. In the citric acid cycle, we'll first take a look at the reactants and the products in terms of the molecules, and then we'll look at the energy. We have 2 acetyl-CoA that we got from breaking down our 2 pyruvates. So we're looking at this in terms of 1 molecule of glucose. We had 1 molecule of glucose, we had 2 pyruvates. From those 2 pyruvates, we made 2 acetyl-CoA. That's what we're sending into the citric acid cycle. So that's a reactant. And we're going to end up with, at the end of this cycle, 4 carbon dioxides. Remember, each one of these is 2 carbons. The idea with cycles is there's an acceptor molecule here that will pick up this molecule. But then at the end of the cycle, we will regenerate that same molecule. So all of the different molecules that we see here, we're not losing any of the carbons from those molecules because we're just rearranging them and getting them back again. So the only thing that can come out of here is what we put in. So we put in 2 carbons here, we can get 2 carbon dioxides out over here. Again, remember we're putting in 2 acetyl-CoA, and that's why we have 4 carbon dioxides here. For every glucose, we had 2 acetyl-CoAs, meaning we'll have 4 CO2s for each molecule of glucose. Let's take a look at the energy gain. Once again, we're putting 2 of these in, so we end up with 4 NADHs right here and 2 NADHs over here. So we have a total of 6 NADHs that we make through the citric acid cycle. We will also make 2 FADHs. And we will make 2 ATPs that we see here. These all represent energy gain. Recall that the NADHs and FADHs will later be sent to electron transport, where we will convert that energy into ATP. Let's take a look at a video that gives us an overview of the citric acid cycle.
Aerobic respiration is the most important process used to produce energy for a cell. It involves several pathways, including glycolysis, pyruvate oxidation, the citric acid cycle, and electron transport. Various sugars, fats, and proteins enter these pathways, and they can all be broken down to produce ATP energy for a cell. In this animation, we will focus on the citric acid cycle, also called the tricarboxylic acid, TCA cycle, or Krebs cycle. The citric acid cycle takes place in the matrix, or fluid, of the mitochondrion. This is where mitochondrial DNA is found and where fatty acid breakdown takes place. The citric acid cycle involves 8 chemical reactions that use acetyl-CoA and oxaloacetate to produce carbon dioxide, NADH, ATP, and FADH2. The NADH and FADH2 are electron carriers that can be used in the electron transport chain to make more ATP for a cell. Let's take a look at the major steps of the citric acid cycle. In the first step of the cycle, a 2-carbon molecule and a 4-carbon molecule are combined to form a 6-carbon molecule. That 6-carbon molecule undergoes multiple biochemical changes during the cycle, and at the end, the original 4-carbon molecule is produced. Each time a carbon molecule loses one carbon, a carbon dioxide is released. So 2 carbon dioxides are formed during the conversion of the 6-carbon molecule back to the 4-carbon molecule. So where does the 2-carbon molecule come from to begin the citric acid cycle? This molecule, acetyl-CoA, is made from pyruvate. Pyruvate is a product of glycolysis, the other major ATP-producing cycle in the cell. Pyruvate is transported into the mitochondrial matrix, where it is oxidized to acetyl-CoA by pyruvate dehydrogenase. This step produces one NADH and one carbon dioxide for each of the 2 pyruvate molecules made from glucose. The acetyl-CoAs are the starting point for the citric acid cycle. In the citric acid cycle, the acetyl group from acetyl-CoA is transferred to oxaloacetate to form citrate. 4 different enzymatic reactions then lead to the formation of succinate. These steps produce 2 carbon dioxides, 2 NADHs, and 1 ATP. Succinate is then recycled back to oxaloacetate through 3 more reactions. These steps produce FADH2 and 1 more NADH. All of the energy molecules made in the citric acid cycle, along with those produced by glycolysis, are essential for fully functioning cells, as errors in these pathways can lead to life-threatening diseases.
Let's take a look at the details of the citric acid cycle. The citric acid cycle consists of a series of 8 reactions, and it is a cycle in that the acceptor molecule is regenerated at the end of this series of reactions and is ready to accept another molecule. So this cycle can continue to run. So our first acceptor molecule in the cycle is oxaloacetate. We will attach the 2-carbon group off of acetyl-CoA, the acetyl group, to oxaloacetate, which is a 4-carbon compound. That will create a 6-carbon compound, citrate. Citrate is known as the first stable intermediate molecule in this cycle, and that's how we name cycles, is for that first stable intermediate molecule. So that's why this is called the citric acid cycle, because citrate is also citric acid. Then we will take the citrate and we will do an isomerization reaction to form isocitrate. Isocitrate will then be converted to alpha-ketoglutarate, and in step 3 here and then in step 4 we lose carbon dioxide, and that's what we see coming off here. So we are going to take off one of the carbons in the form of carbon dioxide, and we see carbon dioxide leaving here, and then we also take off another one over here and we see another carbon dioxide. So recall that citrate was a 6-carbon compound, isocitrate was also a 6-carbon compound, alpha-ketoglutarate is a 5-carbon compound, so we lost one of the carbons, and then succinyl-CoA over here is a 4-carbon compound. So when we went from alpha-ketoglutarate to succinyl-CoA, we end up losing another CO2. So we went from a 4-carbon oxaloacetate, we attached 2 to get a 6-carbon compound. We rearranged to form an isomer, so it's still 6 carbons. We take off a carbon, making a 5-carbon compound. We take off another carbon, making a 4-carbon compound. So down here we're left with 4 carbons. Now we're not going to get rid of any more carbons because remember our oxaloacetate has to be regenerated and it's a 4-carbon compound. So from here on up, after step 4, we're not going to remove any more CO2s. And recall that when we run glycolysis, if we were looking at 1 molecule of glucose, we would have actually had 2 acetyl-CoAs here. So we would have 2 oxaloacetates sitting here, 2 citrates, and that means we would also have 2 CO2s coming off over here and 2 CO2s down there. So we would really have a total of 4 CO2s if we were looking at a molecule of glucose. So once again, remember this basically turns twice if we're looking at a molecule of glucose. After we strip off the last carbon dioxide and we're left with succinyl-CoA, really the remaining steps here, we're sort of regenerating that oxaloacetate. We're rearranging the atoms again to get our oxaloacetate back out. In the process, we also will form ATP. We will form FADH, which is an electron carrier, and up here we'll form another NADH. So if we were to go and look at how much NADH we're getting for each acetyl-CoA, we have 1 here, 2, and 3. But recall that this, if we were doing this for glucose, we would run this twice. So we would actually have 6 NADHs, 2, 4, and 6. And then we would have 2 FADHs if it was a molecule of glucose. And we would have 2 ATPs. We would also have 4 CO2s over here. Now let's take a look at some of the enzymes that run these reactions. So the first reaction, the attachment of the acetyl group onto oxaloacetate, is run by an enzyme called citrate synthase, because when we attach the acetyl group to oxaloacetate, we form citrate. Our second enzyme in this reaction, the one that takes citrate and goes to isocitrate, is called aconitase. Going from isocitrate to alpha-ketoglutarate, we have isocitrate dehydrogenase, which makes sense because we're pulling off a hydrogen. Going from alpha-ketoglutarate to succinyl-CoA, we have ketoglutarate dehydrogenase. Going from succinyl-CoA over to succinate, we have succinyl-CoA synthetase. Going from succinate to fumarate, we have succinate dehydrogenase. Then going from fumarate up to malate, we have fumarase. And then going from malate over to oxaloacetate, we have malate dehydrogenase. Let's take a look at some videos that animate this for us.
The citric acid cycle is a series of chemical reactions that takes place in the mitochondrial matrix. This cycle uses acetyl-CoA derived from sugar and fat breakdown to form ATP, NADH, FADH2, and carbon dioxide. The NADH and FADH2 can be used to form additional ATP through the electron transport chain. The citric acid cycle goes by many names, including the tricarboxylic acid, or TCA cycle, and the Krebs cycle. Citric acid refers to the citrate that is produced in the first step of the pathway. The tricarboxylic acid title gets its name from the 3 carbon dioxides that are produced for each fully oxidized pyruvate. Krebs refers to Hans Adolf Krebs, who identified the full cycle in 1937. He was eventually awarded the Nobel Prize for Physiology or Medicine in 1953 for his discovery. In the first step of the cycle, an enzyme called citrate synthase joins the 2-carbon acetyl group from acetyl-CoA with the 4-carbon oxaloacetate to form a 6-carbon citrate. In step 2, an enzyme called aconitase converts citrate into isocitrate. Next, an isocitrate dehydrogenase enzyme oxidizes isocitrate, a 6-carbon molecule, into a 5-carbon alpha-ketoglutarate. The carbon that was lost is released as carbon dioxide, and 1 NADH is also formed. The carbon dioxide that is released was originally part of oxaloacetate and not acetyl-CoA. In the fourth step, an enzyme called alpha-ketoglutarate dehydrogenase converts alpha-ketoglutarate into a 4-carbon succinyl-CoA. Similar to step 3, this reaction produces 1 carbon dioxide and 1 NADH. In step 5, a succinyl-CoA synthetase enzyme converts succinyl-CoA into succinate. This produces GTP, which is converted to ATP. In step 6, an enzyme called succinate dehydrogenase converts succinate into fumarate. This step makes 1 FADH2. In step 7, a fumarate hydratase enzyme then converts fumarate into malate. In the final step of the citric acid cycle, a malate dehydrogenase enzyme converts malate back to oxaloacetate. Like all steps involving a dehydrogenase, a coenzyme is produced. Here, it is NADH. The oxaloacetate that was regenerated through the citric acid cycle is now ready to join with another acetyl group and begin the cycle a second time. For every 1 glucose that is broken down through glycolysis, 2 pyruvates will be produced. These 2 pyruvates will produce 2 acetyl-CoAs. So for every 1 glucose, 2 acetyl-CoAs will be made and 2 turns of the citric acid cycle will occur. This means each product of the cycle must be doubled. A total of 4 carbon dioxides, 6 NADHs, 2 FADH2s, and 2 ATPs are made through the citric acid cycle. NADH and FADH2 are electron carriers that can produce more ATP later in aerobic respiration. In addition to sugars like glucose, proteins and fats can also provide carbon substrates to fuel the citric acid cycle. Proteins can be broken down into individual amino acids such as alanine, aspartate, and glutamate, and converted into intermediates in the cycle. Fatty acids can be broken down into acetyl-CoA, which then begins the citric acid cycle. This metabolism of sugars, proteins, and fats through the citric acid cycle provides the vital energy necessary to maintain many cellular processes.
The origin of energy production and biosynthesis inside your living cells is the dynamic mitochondria organelle. Mitochondria are filled with metabolic enzymes that catalyze a loop of chemical reactions called the citric acid cycle. The 8 steps of the citric acid cycle gradually break apart 2 carbon atoms from glycolysis, capturing liberated electrons for the electron transport chain, while generating carbon dioxide as waste. The citric acid cycle starts with the end product of a previous cycle, the 4-carbon molecule oxaloacetate. New carbon enters from glycolysis in the form of acetyl-CoA, a 2-carbon acetyl group attached to coenzyme A. Step 1 is the catalytic transfer of a 2-carbon acetyl group from coenzyme A to 4-carbon oxaloacetate, creating 6-carbon citric acid, the molecule that gives the cycle its name. Citric acid is used by your cells for the biosynthesis of fatty acids, lipids, and cholesterol, and is also the substrate for step 2 of the citric acid cycle. The enzyme reaction of step 2 makes a small change to the citric acid molecule, moving the position of an oxygen atom, converting citrate into isocitrate. Step 3 of the cycle removes a carbon atom, forming carbon dioxide as waste, while converting 6-carbon isocitrate to 5-carbon ketoglutarate. During this process, chemical energy is harvested when 2 electrons are transferred to coenzyme NADH, which delivers them to nearby enzymes of the electron transport chain. Step 4 of the citric acid cycle is performed by a huge multi-enzyme complex, connecting multiple chemical reactions with flexible tethers, efficiently moving reactants between active sites, and diverting electrons between pathways. The large complex is made with a repeating unit of 3 core enzymes, working in sequence to remove a carbon atom from ketoglutarate, generating carbon dioxide waste, then attaching 4-carbon succinyl to coenzyme A. The electron from ketoglutarate oxidation is retained by the tethered enzyme before passing it to coenzyme NADH, which delivers electrons to the electron transport chain. Enzymes of step 5 separate succinyl from coenzyme A by breaking an energetic bond, providing enough energy to create GTP. GTP is a building block of RNA and an energy source for protein synthesis. In the most direct path to ATP from the citric acid cycle, other enzymes in the matrix can use GTP to make the chemical fuel ATP. Step 6 is performed by an enzyme embedded in the inner mitochondrial membrane. This enzyme is also known as complex 2 of the electron transport chain. The enzyme catalyzes the oxidation of succinate to create fumarate, releasing 2 electrons that hop through the interior to coenzyme Q, which is reduced. Coenzyme Q travels in the membrane carrying the electrons to nearby enzymes of the electron transport chain. Step 7 takes fumarate, generated by step 6, and reacts it with water to create 4-carbon malate. The final step of the citric acid cycle uses malate to regenerate oxaloacetate, transferring electrons to coenzyme NADH, which supplies the electron transport chain. Oxaloacetate can return to step 1 for another loop of the cycle, and is also an essential building block for making amino acids and the genetic code of DNA and RNA. At the center of cellular metabolism, the citric acid cycle generates both chemical energy and material for biosynthesis of the entire diversity of molecules found inside a living cell.